in our lab notebook that's out in front of us. The reactions of cell respiration and cellular respiration. A date is always helpful. Here's the basic version that I think you guys know about cell respiration. So same with photosynthesis. This is what I think you know. We're going to just advance that knowledge a little bit more. So if you want to draw this little guy in your margin of your paper somewhere, just so you have, food goes in, oxygen goes in, water and carbon dioxide are waste products. They come out. If you're not drawing a little picture, you could draw a horse or a cat or a worm or any other kind of animal. Because all animals do cell respiration. Guess what doesn't do cell respiration? Plants. Nice job. So I'm going to skip a few important steps where you take in protein and lipids and carbohydrates. Those all get broken down by your stomach and different systems do it a different way. We're going to kind of skip to the part where it's already glucose. Okay? So, cells use 2 ATPs to break apart glucose into 2 pyruvates, which produces 4 ATP. So I'll write it all down for you, don't you worry. Break glucose. Pyruvate, one of my favorite words in life. This is our step 1.
Wait, Miss Lehman, how do you pronounce that?
Pyruvate.
Pyruvate?
Pyruvate, yes. Don't worry, there is not a pronunciation test on the midterm. 4 ATP. Step 1. Many little simple cells, like bacteria, or yeast, or other single-celled organisms, are perfectly happy just doing this, using 2 ATP to get 4 ATP. This step has a special name. Walk over here and find my other markers. It's called glycolysis. Glycolysis is the name of step 1. Okay? And I don't, you gotta call it glycolysis or glycolysis, because glyco is sugar and lysis is break. So hopefully an easy way to remember this step is that we break glucose, we break sugar. Glycolysis. No, I'm saying it wrong. Thank you.
Just make the best joke in the world. You didn't hear that?
Nope, I'm not listening. I'm in teaching mode. Thank you. Now, if you drew your little dude in the margin, maybe under him in the margin somewhere, you might want to draw this little data table. Because we're going to track how much ATP we make in this whole process. ATP is kind of super important. You know what? For Elijah, I'm going to highlight ATP energy. I spent some time debating on how much I wanted to tell you guys about ATP. And I think it's enough for us to just know the definition. ATP. All I want you guys to know is that it's a chemical compound that stores and releases energy for our cells. So in this first step, cells have to use 2 ATP, or subtract, -2, in order to produce 4. So how many in stage 1 glycolysis do we actually get?
2.
You had it! 2. Because we use 2, that means subtract. And we produce 4. So it's basically like saying -2 plus 4. If you're in any kind of basic math class. Basic math is that we get 2 whole ATP. Again, little single-celled organisms are totally fine with that. They just need, all they need is a little bit of ATP to help them find more food, to make more ATP, and to produce DNA, and whatever else they have to do. Us, on the other hand, we got a lot more to do. Still holding a pencil, need to take things. Now, here's where we get onto a little bit of a divergent pathway. We either have an anaerobic pathway or an aerobic pathway. Now, if you're labeling this to keep everything straight, step 1 and 2A is the anaerobic pathway. An is Latin for without, meaning none, and aerobic is for oxygen. So this is our, if there's no oxygen available, this is what happens. If you're doing a picture, I think it'll take about 30 a page. Here's pretty much all I'm going to write. pyruvate and gives us a little bit of ATP in the cytoplasm of the cell, not in the mitochondria, not in the powerhouse of the cell. You'll know what I'm talking about a little bit. If you have done any kind of exercise or working out at any point and been sore and sad like me, if you work out for more than 2 minutes, you're like, oh, what? It's because there's no oxygen available. No oxygen. Cells have to do fermentation. Lactic acid fermentation specifically for us. These two steps, if we don't have oxygen available, we'll just cycle between steps. We'll do 1, 2, 1, 2. Little bacteria do this. And some types of bacteria in yeast will do alcoholic fermentation. Which, yes, please don't start a huge discussion about it. That is what makes alcohol for us. We use different bacteria and single-celled organisms to do alcoholic fermentation, which causes us to get beer and vodka and other things like that you can experience when you're 21 or older. Lactic acid is the byproduct, is the waste product of fermentation in our bodies. So, like I was saying earlier, if you work out for more than 2 minutes, like me, and your quads or your arms start to hurt, that's a buildup of lactic acid in your system. You get it. Like when you're getting real swole and you've done like 10 clean jerk things, and you're like, okay, I'm getting the 11th one, but I'm super sore. And you get the 11th one, you're like, it burns. Drink lots of water and massage the area. It'll break up all that lactic acid. That'll let you get it. Anyway, this is just for your reference. If you don't have enough oxygen, like if you're holding your breath while you're doing your yoga positions or something, you're not getting enough oxygen in your system, and your body has to do the lactic acid fermentation. You still are only going to get 2 ATP out of that, which sucks. That's why you get tired when you hold your breath from exercising. Yeah, buddy. So, do it. If you don't have oxygen, lactic acid fermentation. That's all I'm going to tell you about that. Any more details? Little basic cells do this, like bacteria, yeast, viruses will do this because that's all they need. But for us, if there is oxygen, we do something a little more complicated. Oxygen available. Write your question down if you have one. Pyruvate that we made in step 1. Converted to big fun word, acetyl-CoA. Converted into acetyl-CoA. No. Please don't make me explain any further than that because it's already pretty complicated. Start writing some things down. Okay, so now we're getting into the aerobic steps. So anything 2B through 6 is our aerobic. Naveed, get your butt in this classroom.
Naveed, come here.
So 2B through 6. I'm going to talk about 6 steps here. These are aerobic. If there's oxygen available, things can go forward and we can get a whole boatload of ATP for ourselves. Alright? Pyruvates are converted into acetyl-CoA. So here, I apologize, there's not really an easier way for me to say acetyl-CoA. I can tell you more about what that is, or you can look up some of the videos I post later if you want to know more about what a coenzyme is. If not, just write that down. That's more than enough for us. Okay? Still having things happen in the cytoplasm, not in the mitochondria just yet. But we'll get there. We have oxygen available. The pyruvates are turned into acetyl-CoA. From there, that fancy acetyl-CoA enters the Krebs cycle. Krebs cycle is just what we call it because that's who discovered it. Old Krebs. And makes citric acid. Crazy story. The Krebs cycle is also called the citric acid cycle. For obvious reasons, I hope. With that, this is the detail we're going to write about for the Krebs cycle. Right there. We're going to talk about a couple things that come out that we need, but otherwise, we're going to stop right there with detail. If you look in the textbook, at any point you open the textbook, and you see the Krebs cycle all written out. It's super intense. Very complex. Once we have citric acid, it's going to go through a series of reactions, and that's all we're going to talk about. It's actually a big, complex series that it turns into this chemical, and then that, and then this, and then that, and so on. It cycles around. So what's really important for us, just know it goes through a cycle and it produces some stuff that we need for later steps. Here's how it works. Citric acid goes through a series. Shorthand however you want. Series of reactions. Like I write RXN for reactions. Or through. I'm not reading or spelling or anything like that. Series of reactions that produce a couple different things. Produces carbon dioxide as a waste product. Definitely important to know that. It makes 2 ATPs. Not a whole lot, but getting something out of it. And some really cool molecules that we'll just know by their abbreviations. NADH. And hopefully you'll know a little bit more than I did back in high school. And FADH. As you're writing, we'll just review. We have to break glucose into pyruvate. Those pyruvates are then turned into acetyl-CoA. Acetyl-CoA is turned into citric acid. Citric acid goes through a pile of reactions to make CO2 as a waste product. We don't need CO2. That's why we breathe it out. But there's some organism that does need CO2. Plants. We get a couple ATP. We should probably write that down on our little data table over here. Krebs cycle. Krebs cycle produces 2 whole ATP. Well, so far, steps 1 through 4, we don't have a lot of ATP produced, but that doesn't matter because we've made NADH and FADH too. These bad boys are pretty important. In the margin. So you're probably right, you've got your little doodle here, you've got your data table. In the margin next to number 4, it'd be a really good idea to write this down. What FADH and NADH are. They are basically just high energy electron carriers. Super excitable electrons that we pull out of these. That's why we have the Krebs cycle, is to produce those. Almost done. I certainly appreciate your patience and your continued interest in science. So, once we have those, we have ATPs, that kind of helps us keep the cycle going. We have CO2 as a byproduct. We have these bad boys of science, bad boys of the Krebs cycle, NADH, FADH2. Those donate electrons and hydrogen ions. I'll write it, let's see, donate electrons to, I'm going to write it as ETC. Who remembers what that is? I have it over there. So if you're not going to remember ETC as electron transport chain, make sure you scribble that down somewhere too. So they donate electrons to the transport chain and hydrogen ions. We needed hydrogen ions back in photosynthesis as well. Why? For creating a hydrogen ion gradient. For creating, I'll say it as this, creating a concentration gradient. These bad boys donate their electrons to the transport chain. And this is all happening in the mitochondria. So the electrons go through here to facilitate making that hydrogen ion concentration gradient. It's important for this last step that maybe is familiar. Anybody remember how the hydrogen ions work for this step? Or does anybody remember the name of this little guy right here? You would have seen it in the photosynthesis stuff. Electrons go through the electron transport chain, create a protein gradient or proton gradient, and then those protons shoot through this ATP synthase to create a whole boatload of ATP. I have to walk all the way over here, grab my red marker, highlight it for line shot. So that's our last step. This is where we get all the ATP. All of these steps condensed have been working for. So ATP synthase. Synth means make. ACE means protein. So ATP synthase is just a protein that makes ATP. Let me write down the last step. Okay. ATP synthase uses the gradient and electron transport chain to make two things. It makes T, you had it T.
ATE.
Spelling is hard. Do you know what it stands for? Adenosine triphosphate. Good try, but I think it's not super important. And okay, so this last step, this process uses the gradient and the electron transport chain to make ATP, which we need, and water, which is waste. Wrapping up our little data table over here. Drawing and writing step 6.
Let me get
20. Everything we need, so 1 through 4, 5 really, leads us to the point where we can use the ATP synthase to make a whole pile of ATP. We're making a ton of ATP. Given we're only starting with one glucose molecule, we've actually made a decent amount of ATP. If anybody knows the exact number that I'm going to write right here, I'll give you that many extra credit points.
4. Close enough. First try.
Alright, so now this is up to 38. This is a highly efficient volleyball player's cells would make 38 ATP. So it just depends on the age of the cell and how efficiently it's using oxygen. The more you work out, the better your body gets at doing that. For the purpose of the video, I'm going to review one time, and then we'll be done. Do you want to read these all back through?
Not really.
Okay. So, we need a little bit of ATP to break glucose into pyruvates. That gives us some ATP. If there's no oxygen, we do fermentation. If there is oxygen, it turns into acetyl-CoA, which turns into citric acid. It goes through some steps to give us a little ATP, some waste carbon dioxide, these bad boys of science, FADH and NADH, which then donate some electrons and hydrogen ions to the electron transport chain that power the ATP synthase pump to make up to 34 ATP, giving us a total of potentially, if you're a healthy human being like us, 38 ATP.